So, actually getting into the topic of gene duplication this time. Gene duplications allow for modifications to be made to a new gene. So, think you have an original gene that's serving a function, you duplicate that gene, and then you've got two copies of it. The original one's going to continue for doing its usual function, but we have a new copy of the gene and we can kind of edit it a bit, and it can take on a new function. So while the original gene retains its old function, the duplicated gene can assume new duplication results in the creation of gene families. Duplication results in paralogous genes, while genes that share a common ancestor, typically through speciation, are called orthologs. There is something to consider called the C-value paradox, which refers to the disparity between genome size and the organismal complexity. There is no correlation to genome size and the complexity of an organism. This is because many organisms can have many duplicated genes, and duplications occur by the process of unequal crossing over, so kind of errors in the kind of chiasmata formation in meiosis. Retropositioning, where a gene can be reverse transcribed and then reintegrated into the genome, and then you've also got segmental duplication. For retropositioning, that's kind of similar to replication slippage. Retrotransposons code for the reverse transcriptase, which allows retropositioning to happen, and these retrotransposons are easy to spot because they lack an adenine-rich 3' region, which most genes have. Duplicated genes can end up performing subfunctions or even neofunctions. Non-functional duplicate genes, which we call pseudogenes, they just accumulate mutations that factor the normal genes, but they don't do anything. They're typically the result of a frame shift or a premature stop codon. So when a gene is duplicated, it can either form a subfunction, it can be edited to form a neofunction, which is a new function, or if the duplicated gene becomes dysfunctional, then it's just a pseudogene. It doesn't really do anything, but it still accumulates mutations, and we can have a look at them if we're going to kind of try and make a phylogenetic tree or something. So that's talking about gene duplication. We're going to talk a bit more about gene duplication now, and then the idea of entire genome duplication. So like we talked about here, if a duplicated gene assumes a subfunction, then both the purpose of the original gene is being maintained through the original gene and the duplicated gene being expressed simultaneously to maintain that phenotype. So this can happen if a gene kind of gets split and different polypeptides are put into kind of different areas of the genome, but they're still expressed together to produce the same kind of protein or something. But a neofunction is where the duplicate gene has a new function entirely separate to the original gene that it came from. So snake venoms tend to evolve from body duplicate genes, and these genes accumulate mutations, and they are able to assume a neofunction that is completely different to whatever it was doing in the body to form a toxin that helps the snakes kill things or defend themselves. So that's gene duplication. For the genome, we need to know that genotypes are visualized with a thing called a karyotype, which shows every chromosome in an organism's cells. Entire genomes can be duplicated through non-disjunction, through polyspermy, or through hybridization. So non-disjunction is just where in meiosis segregation is done correctly. Sorry, it's not done correctly, and the daughter cells have different numbers of chromosomes. And then polyspermy is when the negative charge of the egg, or the secondary oocyte in the female, can't be maintained, so multiple sperm are able to fertilize the egg at once, resulting in the embryo becoming either triploid or tetraploid or whatever is really possible. Most of the time, they're not really viable though. So more on the genome duplication, there are energetic costs to genome duplication, and each chromosome has a specific region in the nucleus where it resides. So why are genome duplications not selected out, and what use are they? A scientist called Ono suggested that 2 genome duplications occurred early in vertebrate evolution, so theoretically we should have 4 times as many homeobox genes than invertebrates have in a thing called the 1-4 rule. We see this when we observe the Hox genes. Drosophila have a single Hox gene cluster, while vertebrates have 4. So if you look at the diagram on the right here, you can see the Hox gene cluster for Drosophila here, and then the Hox gene clusters for humans here. Some of the Hox genes in the cluster have been lost, but you can see, for example, in this region at the very end of the human embryo Hox gene cluster, there are 4, while in the Drosophila there are only 1, which is evidence of these 2 genome duplications in the early evolution of vertebrates. It's theorized that these vertebrate genome duplications were responsible for providing us with the genetic material we needed to innovate and diversify as a group, but this may not be true because the lobe-finned fish, the Sarcopteridians, remained fairly primitive for millions of years after their genomes had actually duplicated. So basically, genome duplications, because they give us so many extra genes, and those genes are all potentially able to form neofunctions, it's hypothesized that the genome duplications allow us to kind of better diversify as a group and assume new functions, and we're able to adapt all these new extra genes that we have to form new functions so we can do more things. Whether that's true or not is to be debated, but there are some energetic costs to entire genome duplications like we talked about a minute ago, because basically it's very energetically costly, because you basically have to put more resources into replicating more DNA. So, you know, the division of cells is a more energetically costly process, and it also requires cells to be a lot larger because they have to fit a lot more DNA in them from the genome duplications. But if it wasn't selected out, there might be some kind of evolutionary purpose to these genome duplications that have benefited us. Humans genetically engineer oysters and salmon to be triploid for some of these kind of reasons, not only so they're not really able to mate because triploid organisms tend to be infertile, like we said before, more DNA from genome duplication, so they have 3 copies of each chromosome, whereas a normal individual will only have 2 copies of each chromosome. It requires more DNA into their cells, so they need larger cells. So these GMO organisms tend to be larger, and also in the case of oysters, the fact that they're made infertile is actually beneficial to consumers because they taste a lot better when they're not in their kind of mating season, because if they were fertile, a lot of oysters would have a lot of eggs in them, which apparently are not very nice to eat, so they're not very palatable. But by making them infertile and triploid, we're able to avoid them producing eggs, so we can basically harvest and eat them all year round.